This final video involves the physiology of digestion and absorption. So just to recap, digestion, and let's throw in some terms from A&P Part 1. Digestion is catabolic, meaning things are being broken down from large macromolecules into simple components or monomers. We need it small enough for this material to get absorbed into our tissue. So enzymes carry out, if you might recall, hydrolysis. That means breaking chemical bonds by adding water. So water's added to break these bonds. So there's digestion. We're breaking things down. And then once material is breaking down, it gets absorbed. And this happens in our intestine, mainly small intestine. We see the lumen, the inside of the small intestine, which is part of our tract, get absorbed. There's our microvilli brush border where absorption occurs into our cells. These are typically enterocytes in our small intestine. Of course, these cells are separated by tight junctions. You can see it right there, a tight junction, so material can't slip in between. They need to get absorbed into our cells and then out the other side of our cells into our interstitial fluid, our tissue fluid, which will then enter either the blood or lacteals. Will go through a process that involves lacteals, which is part of the lymphatic system and fats. So we need to go through this process here, especially absorption, for each of our four nutrients, for carbohydrates, lipids, proteins, and nucleic acids, because each of the four have just a special way and a unique way of absorbing into our vessels. So beginning with carbohydrates, more than half of our carbohydrates that we take in are digestible and involves starch. And the rest would be indigestible carbohydrates, which provide fiber. So the important point here is that only monosaccharides, the simple units of carbohydrates, can be absorbed. So digestion has to break down our digestible carbohydrates into absorbable monosaccharides or simple units. And we can see here digestion of carbohydrates actually begins in the mouth with salivary amylase, and then our pancreas will secrete pancreatic amylase to complete the digestion into our monosaccharides. Now, glucose and galactose, those monosaccharides, are absorbed via co-transport, so we need sodium ions to enter our cells, our enterocytes, with our monosaccharides. We call that co-transport because they carry each other in. So fructose uses facilitated diffusion, so there's some type of protein in the membrane, in the plasma membrane of the enterocyte, that helps bring in fructose. So just depending on the monosaccharide, it will enter our cells either one way or the other. So all of our monosaccharides leave the epithelial cells through diffusion or facilitated diffusion. So we're talking about right here, they've entered either through facilitated diffusion or co-transport with sodium ions, and then they'll leave the cell through facilitated diffusion into the interstitial fluid. And then we'll see they'll enter the capillary blood in the villi, and then they're transported to the liver, like most of our nutrients are, through the hepatic portal vein, so that this nutrient-rich blood can be processed in the liver before it enters our regular circulation. Lactose intolerance occurs when someone doesn't have enough lactase, which is present at birth, but some people just become deficient in lactase, the enzyme that breaks down the sugar lactose. So therefore, lactose remains undigested. It cannot get to its monosaccharide form, and of course we know the results that you can read here that can come with an intolerance to lactose. So let's go through digestion of proteins. We can see that, well, the building blocks of proteins are amino acid. So we need to break proteins down or digest them into their amino acid components, or close to, at least. So that begins in the stomach with pepsin, hydrochloric acid actually activates pepsinogen into pepsin, which starts breaking down our proteins into smaller and smaller peptides. Peptides are bonded amino acids. And then in the small intestine, the pancreas will release inactive forms of proteases, that we list 3 of them here, and then they become activated into proteases that will break down our proteins into smaller and smaller segments. We also have brush border enzymes on our microvilli listed here, which continue the breakdown process of peptides or small proteins into amino acids. So when we read through this, it's going to sound very similar to carbohydrates now. So amino acids are absorbed through co-transport, again, with sodium ions. But some dipeptides, those are 2 amino acids that are bound together, and sometimes 3 tripeptides, can be absorbed. So they don't necessarily have to go all the way down to their monomers amino acids. But these dipeptides or tripeptides get co-transported with hydrogen ions and hydrolyzed into amino acids inside the cells. So infrequently, transcytosis of small peptides occurred, but it's usually amino acids by the time it leaves the enterocytes. So amino acids leave those enterocytes by facilitated diffusion, just like carbs, enter the capillary blood in the villi, just like carbs, and are transported to the liver via the hepatic portal vein. So we can go back to that picture. Where was it? There it is. Yes, so we have amino acids entering, co-transport with sodium ions, but some of the larger molecules, the di and tripeptides, will use hydrogen ions as co-transport in. And then in the cells, any amino acids that are still in peptide form will typically get broken down to their amino acids. Then you get facilitated diffusion out of the cell and into the blood to the liver. Digestion in lipids really doesn't happen in the mouth or stomach. As you can see, they're of minor importance. But digestion will occur in the small intestine because the liver and gallbladder will secrete bile or bile salts, which will emulsify the fats, which is breaking it down into smaller segments, and then pancreatic lipases will break them down even further. So let's look at how lipids get absorbed into our system, but we're going to use an image on the next slide. So in our duodenum, bile salts will emulsify our fats or lipids into smaller segments, and then add some pancreatic lipases. Those enzymes will break down our fats even further. So the main fat that we have coming into our system is called triglycerides. So the pancreatic lipases will break down triglycerides into monoglycerides, from 3 glycerides to each individual segment, monoglycerides, and glycerol holding the fatty acids together. So we get these free fatty acids, so basically broken down triglycerides, which are surrounded in this little bubble, which we call a missile formation, thanks to our bile salts. So in these little missles, we have just little small units of fatty acids and small fat molecules. So then they'll just diffuse in to our enterocytes. This is a villus in our small intestine. Inside the cell, we have chylomicron formation. So those broken down units will start reforming again. So we create triglycerides again inside of our cells. So we're building. So this is actually anabolic inside the cells. They're also combined with other simple fats, with some proteins as well, into a chylomicron, which will then get released from the cell through exocytosis. These chylomicrons then enter our tissue fluid, which will then enter our lacteals. So chylomicrons are extruded from the epithelial cells by exocytosis, and they enter the lacteals. This is part of our lymphatic system. Now we call it chyle, C-H-Y-L-E, and that will run through the lymphatic system, through lymph nodes, get filtered, which will ultimately get delivered to the blood. When it comes to nucleic acids, it's pretty simple. They're ingested cells that contain DNA or RNA from other living things. They're absorbed via active transport, but let's look at the digestion. Digestion comes from the pancreatic juice, specifically nucleases, ribonuclease and deoxyribonuclease for RNA and DNA, respectively. So that breaks down the nucleic acids into simple form, and also the brush border enzymes on the microvilli would involve further breakdown. And the process is pretty simple. So these little small units of DNA and RNA enter the intestinal cells by active transport via membrane carriers. So active transport, the cell is expending energy, ATP, to bring that material in. Units are absorbed into the capillary blood and villi and transported to the liver and hepatic portal vein. So same story as other nutrients except lipids or fats. So we use the term malabsorption when there's impaired nutrient absorption, any of the four nutrients that we went through, anything interfering with delivering bile or the pancreatic juice, because those are ultimately the main digestive products that break down our food, and be caused by damage to the mucosa in our tract or by some infection. A common type of malabsorption is a gluten-sensitive enteropathy, for example, celiac disease. It's a genetic malabsorption disease where someone has an immune reaction to gluten, a protein that's contained in most of our grains, where our immune cells will damage our intestinal villi, so it's our immune system damaging our own cells, the brush border, so it's very difficult to absorb through this damaged brush border, especially the gluten, that specific protein.